Hi everybody, it's Mrs. Peachy from Summer School Biology, and I'm going to do a quick tutorial on how to use your microscope because we will be using that a lot over the course of the summer. On the screen you'll see a lot of different parts for the microscope. I am not planning on having you memorize everything there, but you'll become familiar with some of the different parts. So first off, we have the arm of the microscope, which has my hand on it right here. This is important because this is how you're going to lift your microscope up. It's a pretty solid piece and you won't be damaging any of the microscope if you lift it by the arm. And then you want to support it underneath of the base with your other hand. So I expect to see everybody carrying their microscope this way and no other way. Okay? When you're putting the microscopes away after use, just make sure that your slides have been removed, the stage has been lowered completely, it's back on the lowest magnification, and you put the dust cover back on, and then you store it back in its proper location based on its number. So we have arm and we have base. Another thing you'll be using is the eyepiece. This is where you're going to look through to be able to see your image on the bottom of the microscope. This is a compound microscope, and there's another type of microscope that I will show you in a moment that you may also use this semester. One of the features of the compound scope is there are 3 to 4 objective lenses here. The smallest objective lens, if you can see this one right here, the shortest, has the lowest magnification, and you're always going to start with that one. That gives you a wide field of view, but it's not as magnified, but it's easier to find what you're looking for and then focus more on that than it would be to start with something that's very narrow and very magnified. Another thing you're going to notice on the screen here, we have a coarse focus knob and a fine focus knob. When you're on your low power, you can use the coarse focus to get everything in focus. But once you turn those objective lenses to a higher magnification, you should stick with the fine focus, because if this stage here moves up and down too quickly, it can smash into the objective lens, and those are pretty expensive. It can damage those. The bottom of your microscope, there's something called the diaphragm, and then there's a light source, and it depends on the microscope. So the diaphragm actually opens and closes a hole to let in more or less light. Mine that I have here, I don't have a diaphragm on here. Oh yeah, I do. I lied. So it's at the bottom. I don't know how well you can see that, but it's just a series of holes. It looks kind of like an old-fashioned pencil sharpener, and it just lets in more or less light because sometimes it can be too bright to be able to make out things that have like a subtle color contrast. You'll find stage clips on the stage here, and that just holds the slide securely onto it. And some microscopes are pretty fortunate in having something called a mechanical stage, which this one doesn't have, but allows you to move that slide kind of left, right, and back and forth, so you can better position what's under the lens. That's pretty much the main parts of the microscope. As you'll notice from the picture up on the screen here, this is actually a binocular microscope here. It's got 2 lenses, or 2 ocular lenses. And then this one is called a dissecting microscope, sometimes called a stereoscope. And it's meant to be looking at larger things like leaves and flowers, blades of grass, insects, things that are not transparent. When you have a compound microscope like this one, the light source is from the bottom, so anything on the slide has to be see-through, otherwise light wouldn't be able to come up through it to your eyes. A dissecting scope like the one on the screen, light comes from the top down. It illuminates the surface of it. Can't see inside of it though, but it allows us to see kind of more larger items like pond organisms and, like I said, other types of things, more macroscopic type things. All right, so what we're going to do now is we're going to look at some images from this microscope. Mine's kind of fortunate because I have a microscope that attaches to my computer that enables me to project what I'm going to see to you. Okay, so let's see here. Let's try this one more time. Okay, what I'm going to show you now is just a screen that shows my video on it. It's the best I can do, my limited technical ability. But what you'll see here is actually part of a sponge, sea sponge. And this is the lowest magnification. So right now I have this on the low magnification, and what I would want to do is use my coarse focus to get this as clear as I can. And say I want to hone in on this area right there. Now with the mechanical stage, you would be able to kind of center that in your field of view. Without it, you have to kind of use your hands and make very small adjustments. Once you have it kind of centered, again, focus it, and then go up to your next higher magnification. Now you're going to use your fine adjustment and just really gently try to get that into focus. Okay, and you may also have to do some movement. Now every movement you make here is going to be way bigger than it was on the previous magnification. So real small movements to get that. Whoops, see, I went way too far. And now I'm looking around trying to find it. And let's see, I might find that if I have no success here, that I would want to go back down to my lower magnification to find what I was looking for. And that's exactly what I'm going to do. I'm going to go back to that lower magnification, and I'm going to try to get that back into focus where I was a minute ago there. There's that center portion, right? Maybe if it was this portion that I wanted. Oops. Right there. So again, switching to the next higher magnification. I do have it in my field of view, but it's slightly off to the right. So to move it, okay, now it's centered. And then I can go up one more power. Again, really fine-tuning this time. Yeah, and we can kind of get into those, see those clusters of cells in the middle, which is what I was trying to look at before. But again, every time I make a move, it's very exaggerated. It's very difficult to see where I am at this high of a magnification. So that's really the bonus of being at that lower magnification. It makes it so much easier to kind of navigate around on that microscopic level. It also is the bonus of having a mechanical stage, because without it, it's really difficult sometimes to control the motion of your hands, especially if you had too much caffeine. Here we go. Back where we began. Okay. So that's kind of what I wanted to conclude with today. So what I would want to do now is I would want to make sure that I cover up my lens cap. You should have a plastic or a cover that will go over top of that. I would want to make sure my stage is all the way lowered. Remove my slide and make sure that my lowest power objective lens is the one that's lined up. And don't forget to turn off your power source. Cover it up with your dust cover and put it back where it belongs. And that'll be it. If you have any additional questions, you know where to ask. And hopefully that helps you out a little bit with how to use the microscope today.